Welcome, this is the Chemistry, Biology, and Math Revision Hub. Today we are doing the scientific article for Question 8 IL Biology for October 2025. Let's begin with Article 1. Some fruit bats eat up to twice their body weight in sugary mangoes, bananas, or figs every day to not only survive but thrive. Unlike humans, these flying mammals can have an essentially permanent sweet tooth and do not develop some of the negative health consequences such as diabetes. A study published January 9th in the journal Nature Communications found that genetic adaptations have helped keep their sugary diets from becoming harmful. The study could have future implications for treating diabetes, which affects an estimated 38 million Americans, according to the Centers for Disease Control and Prevention, the CDC. It is the 8th leading cause of death in the United States and the leading cause of kidney failure, lower limb amputations, and adult blindness. With diabetes, the human body can produce or detect insulin, leading to problems controlling blood sugar, study co-author and University of California, San Francisco geneticist Nadav Ahituv said in a statement, but fruit bats have a genetic system that controls blood sugar without fail. We'd like to learn from that system to make better insulin or sugar sensing therapies for people. Fruit bats versus insect bats. Every day, fruit bats wake up after about 20 hours of sleep and feast on fruit before returning back to their caves, trees, or human-built structures to roost. To figure out how they can eat so much sugar and thrive, the team in this study focused on how the bat pancreas and kidneys evolved. The pancreas is an abdominal organ that controls blood sugar. Researchers compared the Jamaican fruit bat with an insect-eating bat called the big brown bat. They analyzed the gene expression, which genes were switched on or off, and regulatory DNA that controls gene expression. To do this, the team measured both the gene expression and regulatory DNA present in individual cells. These measurements show which types of cells primarily make up the bat's organs and also how these cells regulate the gene expression that manages their diet. They found that the compositions of the pancreas and kidneys in fruit bats evolved to accommodate their sugary diet. The pancreas had more cells to produce insulin, an essential hormone that tells the body to lower blood sugar. It also had more cells that produce another sugar-regulating hormone called glucagon. The fruit bat kidneys had more cells to trap scarce salts and electrolytes as they filter blood, changes in DNA. Taking a closer look at the genetics behind this, the team saw that the regulatory DNA in those cells had evolved to switch the appropriate genes for fruit metabolism on or off. The insect-eating big brown bats had more cells that break down protein and conserve water, and the gene expression in these cells was calibrated to handle a diet of bugs. The organization of the DNA around the insulin and glucagon genes was very clearly different between the two bat species. Study co-author and Menlo College biologist Wei Gordon said in a statement, "The DNA around genes used to be considered junk, but our data shows that this regulatory DNA likely helps fruit bats react to sudden increases or decreases in blood sugar." While some of the fruit bats' biology resembled what is found in humans with diabetes, the bats are not known to have the same health effects. Even small changes to single letters of DNA make this diet viable for fruit bats, said Gordon. We need to understand high sugar metabolism like this to make progress, helping the 1 in 3 Americans who are pre-diabetic. Studying bats for human health. Bats are one of the most diverse families of mammals, and everything from their immune systems to very particular diets are considered by some scientists to be examples of evolutionary triumph. This study is one of recent examples of how studying bats could have implications for human health, including in cancer research and virus prevention. For this study, Gordon and Ahituv traveled to Belize to participate in an annual batathon, where they took a census of whale bats and field samples. One of the Jamaican fruit bats that they captured at the batathon was used to study sugar metabolism. For me, bats are like superheroes, each one with an amazing superpower, whether it is echolocation, flying, blood sucking without coagulation, or eating fruit and not getting diabetes. Ahituv said, "This kind of work is just the beginning." Moving on to Article 2. After getting bit by a bat bug at a recent conference, Armin Sheben had a literal and figurative itch to study bats. The blood-sucking insect is one of many disease-causing parasites that latch themselves onto the flying mammals. Yet, bats rarely get sick in the same way humans do. Mammalian immune systems evolve fast as species are always challenged with new pathogens in their environment. You need to constantly keep pace with new bad guys that are trying to infect and hurt you, says Shiben, who is a postdoctoral fellow in population genomics at Cold Spring Harbor Laboratory and has since recovered from the bite. And while he has studied the genetic adaptations of several mammals, they pale in comparison to the ones that have given bats the ability to fight off infections so effectively. In a new study published today in the journal Genome Biology and Evolution, Shiben and his team have identified the genes that have contributed to bats' rapidly evolving immune system and their unique ability to evade deadly viruses and even cancer. Understanding how bats survive diseases could inspire new immune treatments for humans and potentially help prevent another pandemic. The authors analyzed the DNA of 15 different bat species to get a clearer picture of how their genes evolved over time. They fully sequenced the genomes of 2 bat species, the Jamaican fruit bat and the Mesoamerican mustached bat, and gathered the other species from pre-existing datasets. They then compared the bat genomes to that of humans, mice, and other cancer-susceptible mammals, focusing their attention on the sequences that encode proteins responsible for causing or preventing diseases. To start, they lined up the homologous genes, or shared genes, among different species inherited from a shared evolutionary ancestor. It's like comparing apples with apples, explains Shiben. With each homologous gene, they hypothesized 2 scenarios: if bats lost it or if it mutated. If the flying mammals completely lost the gene, it suggests that the omission is important in fighting disease, but if it remained with subtle changes in the DNA sequence that are only found in bats, it could show a change in gene function that somehow helps the group stay healthy. In the end, the most striking changes the team detected were in type 1 interferon, or IFN genes, which are important for controlling inflammatory responses to infections. Specifically, they observed a shift in the number of antiviral IFN alpha and IFN omega genes. For instance, 3 bat species seemed to have lost all of their IFN alpha while increasing the number of IFN omega genes. According to Shiben, the most surprising finding was observing the loss of IFN alpha and addition of more IFN omega genes, which hadn't been reported at all before. The results suggest the new IFN omega and missing IFN alpha genes are important in bats for resisting viral infections while preventing overactive inflammatory responses, a feature that has made inflammation a double-edged sword in humans. But while the findings have put geneticists one step closer to understanding how bats evolved their unique ability to resist cancer and viruses, it doesn't paint a complete picture. The study focuses only on the genetics of innate immunity, the immediate immune response to infected cells, says Tony Shantz, a professor at the Center for Vector-Borne Infectious Diseases at Colorado State University, who was not involved in the study. It does not include information about bats' adaptive immunity, which consists of the antibody and T-cell responses that many mammals use to fight diseases. These are 2 very different but complementary components of immunity, Shantz explains. Nearly all of the focus on bat immunity to date has been on innate immunity, principally because the study of adaptive immunity requires live animals, which few groups have and is much more complicated. Even without a full set of information, understanding the changes in the bat's innate immune system could help scientists develop genetic treatments for humans that decrease susceptibility to certain illnesses. We can also learn which genes drive bats' 20 to 30-year lifespans, or how their bodies have adapted to process sugar-rich foods without developing the negative consequences seen in people with diabetes. And though bats have gained a notorious reputation for their purported role in spreading COVID, Shiben hopes that these new findings could point researchers in the right direction in understanding how the animals host such potent viruses and parasites without getting very sick. One day, he says, that information could be used to prevent our species from suffering major symptoms when infected. It's absolutely not misplaced to believe that studying bats could help us prevent another pandemic. Before we go to the questions, I would like to make a disclaimer. These questions are my own work and are not taken from the official exam paper. I have not seen the paper and I do not know the exact questions that will appear. They are designed only as practice material to help you prepare for your exam. The first set of questions are about glucose regulation, diabetes, and effects on organs. 1. Explain how insulin lowers blood glucose levels. Insulin binds to receptors on target cells, such as muscle and liver, stimulating the uptake of glucose from the blood. In the liver, it promotes the conversion of glucose to glycogen, reducing the concentration of glucose in the bloodstream. As levels fall, insulin secretion decreases, maintaining homeostasis. 2. How does glucagon help maintain blood glucose when levels are low? When blood glucose falls, alpha cells in the pancreas secrete glucagon. This hormone acts on the liver to stimulate the breakdown of glycogen into glucose, which is released into the blood. As levels rise, glucagon secretion is reduced. 3. Describe how negative feedback operates in blood glucose regulation. Negative feedback ensures that changes in glucose levels are detected by the pancreas and corrected. A rising glucose triggers insulin release, which lowers glucose back to normal. A fall in glucose stimulates glucagon release, which raises blood sugar, restoring balance. 4. Why do fruit bats not develop diabetes despite high sugar intake? Fruit bats have evolved pancreases with more insulin and glucagon-producing cells, giving them a greater capacity to regulate blood sugar. In addition, changes in regulatory DNA ensure that the relevant genes are activated rapidly. These adaptations allow them to avoid the damaging effects. 5. Explain why uncontrolled diabetes can lead to blindness. Persistent high glucose levels damage the small blood vessels in the retina. This reduces the supply of oxygen to photoreceptor cells, causing them to degenerate. Over time, this results in impaired vision and can lead to blindness. 6. How does the pancreas of fruit bats differ from insect-eating bats? Fruit bats have pancreases with more cells that produce insulin and glucagon, allowing them to regulate sugar-rich diets. Insect-eating bats instead have pancreases specialized for protein digestion, with fewer insulin and glucagon-producing cells. These differences reflect dietary adaptations. 7. How does insulin perform its action? Insulin is a peptide hormone made from amino acids joined by peptide bonds. It cannot pass through cell membranes and instead binds to receptors on the surface of target cells. This binding triggers internal signaling pathways that alter cell activity. 8. What happens if insulin receptors on body cells fail? Glucose is not taken up effectively into cells, so it remains in the bloodstream. This leads to persistently high blood glucose levels, characteristic of type 2 diabetes. The pancreas may continue producing insulin, but its effect is reduced. 9. Why do fruit bats produce more glucagon-secreting cells than insect bats? With a diet very high in sugar, fruit bats need to prevent dangerous falls in blood glucose once digestion slows. Extra glucagon-producing cells allow rapid release of glucose from glycogen stores. This balances the high insulin secretion that follows feeding. 10. Explain how protein synthesis allows pancreatic cells to produce insulin. The insulin gene is transcribed into mRNA in the nucleus and then translated on ribosomes in the rough endoplasmic reticulum. The resulting polypeptide is folded and modified into active insulin in the Golgi apparatus. It is then packaged into vesicles for secretion by exocytosis. 11. How can diabetes lead to kidney failure? High blood glucose damages the capillaries in the kidneys glomeruli, making them leaky and inefficient. Proteins may be lost in urine, while wastes are not filtered effectively. Over time, this progressive damage causes chronic kidney. 12. Explain how fruit bat kidneys are adapted to their diet. Fruit bats consume large volumes of sugary fruit, leading to high water excretion. Their kidneys contain more cells specialized for trapping salts and electrolytes. This prevents mineral loss and maintains osmotic balance. 13. Describe the process of ultrafiltration in the kidney. Blood enters the glomerulus under high pressure. Small molecules such as water, glucose, and ions are forced through the basement membrane into Bowman's capsule. Larger molecules like proteins and blood cells remain in the bloodstream. 14. Explain the process of selective reabsorption in the kidney. In the proximal tubule, useful substances such as glucose, amino acids, and ions are actively transported back into the blood. Water follows by osmosis, conserving valuable resources. This ensures that only wastes remain in the filtrate. 15. How does ADH affect water reabsorption in the kidney? ADH increases the permeability of the collecting duct to water by stimulating aquaporin insertion. More water is reabsorbed into the bloodstream, producing concentrated urine. When ADH levels fall, less water is reabsorbed and dilute urine is formed. The next set of questions is on gene expression. 16. What is the role of regulatory DNA in gene expression? Regulatory DNA determines when a gene is switched on or off. It controls the timing and level of gene expression, often by binding transcription factors. In fruit bats, changes in this DNA allow precise control of sugar metabolism genes. 17. Describe the difference between epigenetic changes and mutations. Epigenetic changes alter gene expression without changing the DNA sequence, for example by DNA methylation. Mutations involve a permanent change in the DNA sequence itself. Epigenetics can be reversible, whereas mutations are not. 18. What is DNA methylation and how does it affect gene expression? DNA methylation involves the addition of methyl groups to cytosine bases in DNA. This usually prevents transcription factors from binding to the gene, switching it off. It is a reversible form of epigenetic control. 19. How does histone modification influence transcription? When histones are acetylated, the chromatin becomes less tightly packed, allowing transcription factors access to the DNA. Deacetylation reverses this and makes transcription less likely. This provides a mechanism to control which genes are expressed. 20. Suggests why epigenetic control is important in regulating insulin production. Epigenetic changes can ensure that insulin genes are switched on when blood glucose is high. This rapid activation helps maintain homeostasis. In fruit bats, such precise control is vital to manage their sugar-rich diet. 21. What is meant by a gene knockout experiment? In a gene knockout, a particular gene is deliberately disabled or removed. The resulting phenotype is studied to determine the gene's function. This technique helps identify which genes are critical for traits such as blood sugar regulation. 22. Explain how gene silencing differs from gene knockout. Gene silencing prevents a gene from being expressed without permanently altering the DNA. This may involve RNA interference that blocks translation. Unlike knockout, the silencing effect can be temporary or reversible. The next set is about evolution. 23. Why do scientists compare homologous genes across species? Homologous genes are inherited from a common ancestor and usually perform similar functions. Comparing them allows scientists to identify conserved and altered regions. These differences may explain unique adaptations, such as disease resistance in bats. 24. How does natural selection explain the evolution of sugar metabolism in bats? Mutations took place that gave some bats advantageous alleles that enabled them to regulate sugar more effectively. Fruit bats with these selective advantages were more likely to survive and reproduce. Their beneficial alleles were passed to offspring. Over generations, these traits became common in the population. 25. Why might small DNA changes be important in evolution? Even single-base substitutions can alter protein structure or gene regulation. In bats, minor DNA changes in regulatory sequences improve glucose metabolism. Such small changes accumulate to produce significant adaptations. 26. How does the comparison of insect-eating and fruit bats illustrate divergent evolution? Both groups share a common ancestor, but they adapted to very different diets. Fruit bats evolved pancreases with more insulin and glucagon-producing cells, while insect bats developed kidneys and pancreases suited for protein breakdown. These changes reflect divergent evolution driven by different ecological niches. 27. Why can losing a gene sometimes benefit survival? Gene loss may prevent harmful processes such as excessive inflammation. In some bat species, the loss of interferon alpha genes reduces damaging immune responses. This allows them to survive infections that would cause illness in other mammals. The next set is about immunity. 28. What role do interferons play in the immune response? Interferons are proteins released by virus-infected cells. They stimulate neighboring cells to increase their antiviral defenses. This limits the spread of infection in the body. 29. How do bats' interferon genes differ from other mammals? Some bat species have lost interferon alpha genes while expanding interferon omega genes. This unusual pattern provides strong antiviral defense without excessive inflammation. It helps them tolerate infections that make other mammals sick. 30. Why do bats rarely suffer from viral infections despite being hosts? Their immune system responds rapidly through innate mechanisms such as modified interferons. They avoid harmful overinflammation that would normally cause symptoms. This allows them to live with viruses without becoming sick. 31. What is adaptive, specific immunity, and how does it differ from innate, non-specific immunity? Adaptive immunity is a specific response involving lymphocytes, B and T cells, and antibodies. It develops more slowly but provides memory for long-term protection. Innate immunity, by contrast, is immediate, barriers and inflammation, non-specific, and does not provide memory. 32. Why do scientists describe inflammation as a double-edged sword? Inflammation is essential for fighting infection because it recruits immune cells to the site of injury. However, if it becomes excessive or prolonged, it damages healthy tissue and contributes to disease. In humans, this harmful overreaction can worsen conditions such as sepsis or autoimmune disease, whereas bats appear to have evolved mechanisms to avoid such problems. 33. Explain why bats' immune systems are considered an evolutionary advantage. Bats can resist viral infections and tolerate pathogens without developing severe illness. This allows them to occupy ecological niches where they are constantly exposed to viruses. Their unique immune adaptations may also contribute to their unusually long lifespans. 34. Why is adaptive immunity harder to study in bats compared with innate immunity? Adaptive immunity involves complex processes such as antibody production and T-cell activation, which require live animals to study. Very few laboratories maintain live bat colonies, making such work difficult. Most research therefore focuses on innate immunity, which can be studied with DNA and tissue samples. 35. Explain the role of T helper cells in the immune response. T helper cells bind to antigens presented by antigen-presenting cells. They then release cytokines that stimulate B cells to produce antibodies and activate cytotoxic T cells. This coordination ensures an effective specific immune response. 36. Why is the bat immune system considered highly efficient? Bats show rapid innate responses with modifications to interferon genes. They resist viral infection without excessive inflammation. This balance allows them to survive infections that would be fatal to humans. 37. How do antibiotics act against bacteria? Antibiotics target structures such as bacterial cell walls or ribosomes. They kill bacteria or inhibit their growth without harming human cells. This makes them effective treatments for bacterial infections. 38. Explain how antibiotic resistance develops. Random mutations may give bacteria resistance to an antibiotic. When exposed, resistant bacteria survive and reproduce, passing on the allele. Overuse of antibiotics accelerates this natural selection process. Why is antibiotic resistance a major concern for medicine? Resistant infections are harder to treat and require stronger or multiple drugs. This increases treatment time and costs and raises the risk of untreatable superbug infections. The spread of resistance reduces the effectiveness of existing antibiotics. The next set is about DNA sequencing and research methods. 40. Explain the role of PCR in studying bat genes. PCR amplifies specific DNA sequences, producing millions of copies from tiny samples. This allows detailed analysis of genes of interest. It is essential in sequencing and comparative studies. 41. How does gel electrophoresis separate DNA fragments? DNA fragments are placed in a gel and subjected to an electric current. They migrate towards the positive electrode because DNA is negatively charged. Smaller fragments move faster and travel further than larger ones. 42. How are microarrays used to study gene expression? Microarrays contain thousands of DNA probes fixed on a chip. mRNA from cells binds to complementary probes, producing signals. This reveals which genes are active and at what level. 43. What does it mean if a gene is lost in evolution? A lost gene is no longer present in the genome, usually due to deletion or inactivation. If the species thrives without it, the loss may confer an advantage. In bats, loss of interferon alpha genes is linked to better viral tolerance. 44. How do homologous genes support the study of evolutionary relationships? Homologous genes are inherited from a shared ancestor and usually perform similar roles. By comparing these genes across species, scientists can identify where changes have occurred. Such comparisons reveal how different species adapted in response to environmental pressures. 45. Why is sequencing many bat species useful when studying immunity? Analyzing a wide range of bat genomes helps reveal which genetic changes are common across the group. This allows researchers to distinguish between random variation and consistent evolutionary trends. Such comparisons highlight genes most likely responsible for disease resistance. 46. What advantage does whole genome sequencing have over studying single genes? Whole genome sequencing provides a complete picture of an organism's genetic material. It allows scientists to detect unexpected changes, such as gene losses or duplications across the entire genome. This broader approach gives more reliable insights into adaptation. The next set is about applications and human health. 47. How might studying bats help improve diabetes treatment? Understanding how bats avoid diabetes despite a sugary diet could reveal new ways to regulate blood sugar. This knowledge might guide the design of better insulin or glucose sensing therapies. It could reduce the impact of diabetes in humans. 48. Why are bats considered important in medical research beyond diabetes? Bats show resistance to viral infections and certain cancers. Their immune and genetic adaptations may hold keys to new treatments. Studying them provides unique insights not available from other mammals. 49. Explain how studying bats might help prevent future pandemics. Bats carry many viruses without becoming ill. Understanding their tolerance mechanisms could help scientists design therapies that reduce human disease severity. This knowledge could limit the impact of future pandemics. 50. Explain why scientists are interested in bats for cancer research. Bats appear to have a lower incidence of cancer despite long lifespans. Their immune system may suppress tumor development more effectively than in other mammals. Studying these adaptations could inspire new approaches to human cancer therapy. 51. Why might therapies inspired by fruit bats be more effective than current insulin treatments? Current therapies replace or supplement insulin but do not fully mimic natural regulation. Fruit bats have genetic systems that sense and adjust blood sugar with precision. Replicating these mechanisms in humans could provide tighter glucose control. 52. How could stem cell therapy be used to treat diabetes? Stem cells can be induced to differentiate into pancreatic beta cells. These cells would restore insulin production in patients with type 1 diabetes. Successful transplantation could re-establish normal blood glucose regulation. 53. Suggest one limitation of organ transplantation as a treatment for diabetes. A transplanted pancreas may restore insulin production, but the patient risks immune rejection. Long-term use of immunosuppressants is required, which increases susceptibility to infections. This makes transplantation less suitable for widespread treatment. In relation to broader biology themes, 54. How does the concept of trade-offs apply to bats' immune adaptations? Most mammals must balance fighting infections with avoiding excessive inflammation. Bats appear to have shifted their interferon gene profile to reduce harmful inflammation while still resisting viruses. This represents an evolutionary trade-off that benefits their survival. 55. Why is it significant that bats live 20 to 30 years compared to other small mammals? Small mammals usually have short lifespans due to high metabolic rates and disease vulnerability. Bats live much longer, suggesting that their immune and metabolic adaptations reduce damage over time. This exceptional longevity highlights their evolutionary success. 56. How might studying bats change the way scientists view junk DNA? Once thought useless, regulatory DNA has been shown to control key genes in bats. Changes in this DNA allow them to adapt rapidly to sugar-rich diets. This challenges the idea of junk DNA and reveals its evolutionary importance. 57. Why do researchers describe bats as superheroes of evolution? Bats possess extraordinary traits such as echolocation, blood feeding, virus tolerance, and resistance to diabetes. Each of these adaptations has allowed them to thrive in diverse ecological roles. Their range of abilities demonstrates remarkable evolutionary innovation in connection to adult blindness. 58. Describe how rods and cones differ in function. Rods are highly sensitive to light and allow vision in dim conditions, but they do not detect color. Cones are less sensitive but detect red, green, or blue light, enabling color vision. Together, they provide a complete visual picture. 59. Explain how the pupil reflex protects the retina. In bright light, the circular muscles of the iris contract to constrict the pupil. This reduces the amount of light entering the eye, protecting the photoreceptor cells from damage. In dim light, the pupil dilates to maximize light capture. So, this brings us to the end of the video. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.